Hello guys, my name is Dr. Varun and you are watching Chlorophyll Classes only on YouTube. Today is Friday and Friday is meant for 12th class. If you are new to this channel, then I'll let you know that we are preparing for NEET 2026, where else we are even preparing for the board examinations for 12th. And for that, we have made a plan that every Monday, Tuesday would be for the 11th class and every Friday and Thursday would be for the 12th class. Well, to little bit quick up our topic and our syllabus and to complete it as fast as possible so that we can come up with really other things also, we have planned that we will be keeping Monday, Tuesday, Wednesday for 11th class syllabus, whereas Thursday, Friday, Saturday as a 12th class syllabus. And we will try to complete as much as possible. For 12th class, we are still stuck with the genetics, although we have to start this month molecular basis also. That will be started also. Don't worry about it. This comes to be the second last video for this chapter. So let's just dive into it. So the topic that we're going to be studying is the sex determination. In humans, you might have already studied that sex determination will take place which type of sperm, whether the X sperm or the Y sperm goes and matches, and that's how the female or a male child has been formed in human cases. So male sperm is responsible for it. Okay. Now let's just come back let's see how the other organisms do that. So there are different ways which came up which tell us about the sex determination. So one we have already discussed, we will learn it more. Okay, so what it is sex is what? It's an aggregate of characters which morphologically distinct between whether someone is male, means producing the sperm, or the producing the egg, female. So these are statements just putting you, like I am writing these statements. The reason is that because in your examination statement based question comes and then you get confused seeing that fancy words, but we will learn all the fancy statements. Okay, we will make our English also super good because in the medical course it will be studies in English. Okay, so your English should be good enough. Okay, moving on, then what is it? In higher organisms means like your fishes, your reptiles, turtles, amphibians, mammals, birds. These are considered as what? Higher animals. Or higher plants, like what? Like your higher, normally considered as bryophytes, pteridophytes, gymnosperm and angiosperms as their higher plants. Their sexes are morphologically different. Means what? That male and female body is also different. Their look like is also different. So means there might be, if the body is itself different, we call it dioecious case. Whereas in lower plants are considered as algae. Okay, and lower animals like invertebrates, certain of the invertebrates, both the sexes are present in one organism. You can't distinguish that this is a male organism or a female organism. Both the sexes are present, and such bodies are termed as monoecious. We learned this thing. If you have tackling me with the 11th class, you know that what it's mean. Okay, if not, then here I have told you. So this is very important for you to understand monoecious and dioecious. Okay, coming up to the next thing. Now those organisms, even humans, they are what? They have two types of chromosomes present, and that's where two different types of body forms us. Two types of chromosomes. What are they? With terms of sex, chromosomes are two types. Here I am telling you that in terms of chromosomes in our body, we have what? 46 chromosomes, or we can call it 23 pairs. So in them, few are autosomes, few are allosomes. Allosomes are what? Sex chromosomes. Means XY is what? Are sex chromosomes. Means in female XX, in male XY. So this pair of sex chromosomes are there. Rest all the 22 pairs, they are what? Contain as autosomes, which are responsible for the somatic characters. Means my eye color, my hair texture, my skin color, all of those things. Okay, so they come into what? Autosomes. Very important. What is autosome and allosome? You should remember. Moving on to the next thing, that is sex determination. The first way of sex determination is with the help of chromosomes. The first one is XXXY. XXXY is what? That comes in humans, higher organisms like your all these apes, birds and all, they have the similar kind of things. Mammals have the similar kind of thing. Then Drosophila also had this one. So Drosophila is what? genetics jackpot. Okay, so then females are normally homogametic, means their gametes would be what, similar, means all them will be XX, carrying the X chromosome, sex chromosome, whereas male will be heterogametic, means it has two different types of chromosomes. One is androsperm, another is gynosperm. Androsperm is what, having the X, sorry, andro is Y. Andro word itself for the male, so male is for the Y chromosome, and gynosperm for the X. So this is what happens. So this you have already done it in the 10th class, so we are not dipping deep into it. Next is XX and XO. Here what happened, XO means not O, it means O means 0, not present. Okay, it seems in case of cockroach. So what happened here also, the female has what, it has XX. So female has 2X chromosomes, whereas male has what, only 1X chromosome, the other one is absent. And once that condition will come, it will what, it will be male. Okay, so if I had to make it in a like a kind of a thing, what will happen, so see that the female has what, XX, the male has XO. So they will make what, two different types of, like here the gametes will be like this, whereas here the gametes will be what, XO, means gamete absent. Okay, now this comes female form, means a female cockroach will form. This will come, what happened, male cockroach will form, because one X is coming, other one is not even coming. Okay, so this will give what, female cross, this will give what, male cockroach. In short, what is happening, some of the sperm has one chromosome less present. Okay, so one chromosome less sperm, that's it. Okay, I hope this thing is understood to you. Moving up to the next thing, that is ZW or ZZ. Here what happened, here also the female is heterogametic. Now the situation changed, means female will have what, X and W, whereas male is XX. So here the sex determination would be the factor of what, factor of egg, opposite of what is present in humans. Okay, same, this is seen in birds, fishes and certain butterflies. Okay, whereas similar things like XX, XO we have studied, where the male do not have one X chromosome present. Here also what happened, the female has only one Z chromosomes, like some butterfly and grasshopper. This can be asked to you and has been asked again and again. So examples you have to study. I hope this thing is understood to you. Okay, moving on to the next thing, that another way of sex determination is seen in case of honeybee. Now think about honeybee. So honeybee has three different type of organism in his hive. Okay, I'm gonna starting with a story. Okay, so remember that honeybee has three different types of organy in its colony. So this is what, beehive. This is what, beehive. It has three different organisms. One is queen bee, one is drone. Drone is what, males. Okay, which will mate with the queen and give the new progeny. Okay, and another is the workers. They are only what, females. Okay, they are reproductively sterile. Now let's see that what is happening. Now normally the queen is what, queen is has two different types of eggs present. One is fertilized, other is non-fertilized. What is the case with it? Now the queen is there. Imagine that this is the queen. Okay, I'm gonna make a crown also for the queen, because queen slays, right? So queen is there. Queen is what? Queen is a diploid. Okay, now when a diploid organism, because I told you most of the organisms are diploid. Okay, so if the queen is a diploid, it has to make gametes. How it will make its gametes? Eggs. It will make eggs with the help of meiosis. So this is what, multiple eggs the queen made. And all of these eggs are what? N haploid. Now with this egg, what has to come? There has to be coming up of what? Sperm to form. So there will be what? Sperms will come. Who will give us sperms? Drone will give the sperm. Drone is what? Male. It will give sperm. Sperm is also what? N. They will fertilized. Now the problem is that the sperms which are fertilizing this queen bee are very less. Okay, but the eggs are a lot. So what will happen? only few of the queen's egg will be able to fertilized. Okay, so what happened? Two cases happen. Certain eggs get fertilized and form what? Zygote, diploid. A lot of them remain unfertilized. But both develops. Then we're like, Ayo sir, both developed means in their body there is a change in the generation. Let's just talk about what is happening. They develops into what? They develops into drone, male. Don't see me like this, just understand. Whereas these zygotes developed into female. As they developed into female, few of them will be chosen by the queen and fed on royal jelly. And that will make what? Future queen. Whereas other would left out will act like what? Workers. Did you understand this fact? Whereas this drone, now the drone becomes what? Unfertilized egg means haploid, means drone is haploid over here. How it's happening? Means in their body only half of the chromosomes present. So if they had to make the sperm, how they will make it? Think that whenever I say that gametes form, meiosis used to be done, right? To reduce the ploidy level. But here if we reduce it, it will become n/2, which we do not want. So what we will do? So they will do what? They will do simple mitosis and differentiation of the cell. And this differentiated cell will turn into what? Sperms. Understood this thing? So this is about the sex determination in honeybee, easiest thing. Now here the statement comes, that is that these drones do not have father or sons, but have grandfather and grandson. Let me know these things. Okay, so this thing is there. This is the haplodiploidic mechanism of sex determination. Means haploid will be the drone males, diploid would be the females. That is only they want to tell you. Next is what? That the genic balance. In the genic balance theory, what happened? We see that how many X chromosomes are there in terms of their autosomes, okay, or their ploidy content. So it is proposed by G.B. Bridge. It says that Y chromosome has no role in sex determination. It's only the X chromosome and their number that decide them. If someone has 3 X chromosome, so and its ploidy is 2n, okay, diploid. So 3 by 2, that is 1 by 5. They will become what? Meta females, because extra female characters are there. But if someone is XX, means normal female, okay, so it comes 1. It is what? Normal female will form. But in males, what happened? Only one X is present, so XY. So only one X present upon the ploidy is 2n. So they becomes what? 0.5, and 0.5 will become what? Males. But what happens if there are 2 X present, but ploidy is 3n, means one extra autosome is present in their case. Okay, what it is, we will learn it in the mutation topic. So this becomes 2 by 3. And this comes 0.66, and this is what? Intersex case. In reality, beta, this don't look like this. It is actually like this. Okay, they have an extra copy of X. This extra thing that is coming up over here is added up here also. Okay, we will learn this and understood it later. Okay, so when we will be doing some mutation topic, that's how it is happening, what is it is happening. Okay, coming up to the next thing, that is bar body method. It says that when they were studying about cell division and all, they found that a darkly stained body, and they call them what? Bar body. Okay, and that bar body is nothing but the 2X chromosomes. Any extra bar body coming means, like if the 2X chromosomes come means in the female, it is considered as 1 bar body. But sometimes what happen, extra than that is coming up. Okay, if like for example in this case meta females, so what happened? Extra 1X is coming, so 1 extra bar body is there. So this is what bar body is absent in case of males, because in males only one X is present, which will not be represented. 2X will represent 1 and then additional. So now the question 2 is that you have observed this. Okay, how many bar bodies there? How many bar bodies are there? So there is what we have told, that these two will act like one bar body, and every additional will act like one bar body. So this is what 1, 2, 3. So 3 would be the answer to this. Understood? They can ask you this question from this topic. Next comes the environmental sex determination. Now sometimes what happen, environment also influence that what gonna happen. Okay, so very first thing is that temperature fluctuation can change the sex determination in the turtle. So what happened in the Puri, okay, Odisha, what happened? There is what lot of turtle comes and they lay their egg and the parents go back and the later on egg comes out. Now in the egg either the male will come or the female will come. But what happens when the temperature increases due to global warming or anything, the temperatures are increasing. We have seen that in their case more females are being formed, because the temperature influenced, the temperature is more, female's turtle will form. If the temperature is less, then the male will form. Okay, and if the equilibrium, then equal amount will form. Okay, so this thing is there. Same goes with another way where how the environment habitat work. Okay, so in their cases there is what Bonilla worm. Now Bonilla worm, like for example that I am the Bonilla worm. So what happened? I'm the female. Imagine that. Okay, I'm the female. Okay, Pookie female. Okay, so what happened? I lays my larvas, my baby. So my baby, all of them when they are present freely, they are female. But some of my female baby come and goes through my proboscis. Proboscis is what, if you remember about the worms in the hemichordate, what happened? Their body is like this. And these are the gills present. Collar, this is the collar, tail, gills, and proboscis. Proboscis is what? A kind of a excretory structure is present here. Okay, so through this what happens, certain of them goes inside the body, okay, and live like that the larva, my own baby going inside my body through the proboscis and living inside my body as a parasite. And now my body will release hormone for me. Under the influence of those hormones, they change themselves into males and living like a male in my body as a parasite, eating my food and also fertilizing my egg. So think about me as a female, what is happening with me. But the nature is curious. So I always felt that the God is very notorious. He is doing something like this. Okay, the nature is doing something like this. Okay, so this is like one of the funny fact. Moving on to the next thing. With this, our sex determination is over. We are coming up to the next topic, that is variation. Now you will say that, sir, in NCERT variations are not there. Beta, the chapter name itself is variations. We have to study this so that we can say that we haven't missed out any things. Now what is variations? Variations are small changes that are occurring in the body and they sometimes even go unnoticeable. Can be noticed, cannot be the noticed, but they are what? They are the changes in the body of an organism which can make a big change later on. And here comes that there are two types of changes which can actually make the differences in the body. First is somatic variations, other is blastogenic variations. Somatic variations means which are occurring in your somatic cells, somatic chromosomes. Okay, means for example, my ears becomes this large, like elephant ears. This is what somatic variations. But another is the blastogenic variation. Blasto word is coming from the blastula, means it is occurring in the germinal layers. Okay, so if something occur in the germ cells, means your sperm and all those things, then it will be inherited because they are the base cells. On that only the next genome will be copied on, and the difference has come in that cell only. These are the two types. Now these somatic variations will be acquired in the lifetime and they will be died. Okay, for example, I can teach good, maybe my kids won't be able to teach good. Okay, so blastogenic means there can be mutation crossing over, they will be passed on to the next generation for sure. Next, the types of the somatic generation. So they could be environmental, like for example, if I go in the sun, tanning will occur, but my dad is not going, he don't have any tan. Okay, so this is tanning. Then heterophily in the buttercup, you know that when they are in the water and when they are in the terrestrial environment, how different they look like. Okay, then use and disuse of organs. What happens sometimes, if we stop using one organ now, it will be wasted. Okay, but if we'll keep on using it, it becomes better in the every respect. So same thing happens. Okay, so this is being given by Lamarck. It happens, but it's like a not goes to the next generation. Like for example, if a lion is there, it is in the cage, it will be less powerful compared to the lion who is in the forest, because in the cage they are getting easy food, they are not hunting wild and those kind of things. So this thing will be what use and disuse of the organs, that the lion has lost the capability of doing the hunt. So that happens with the caged animals. Even with the birds also, they also forgot to fly. Okay, so they have to learn it all over again, okay, for the survival. Then the conscious effort means like what, sometimes we want to do something and that is a conscious, like studying. You guys are studying right now, so it is what your efforts. Maybe your kids don't study, maybe our parents might not have studied, this thing would have happened. Then coming up to the blastogenic, they are what two types, discontinuous and continuous. Since I told you what blastogenic inheritance will go to the next generation. So now here what can happen with the mutation might be occurring in the this generation, maybe in the baby is not occurring. So they are what substantive or merismatic. Substantive means discontinuous means it might be present in the next generation or might not be present in the next generation. It might skip and then again come back. Okay. So what is what substantive means? Change in the shape, size, color, organs. Like the amount of milk in the cow. Maybe this cow which I have right now is giving me 2 liters of milk. Maybe the next one is giving me only 1 liter. Maybe the next is giving me again 2 liter or 3 liter. Means this change is coming in the yield product. Okay, or the meristem means increase in the organs level, means number of organ increase, like the arms of the starfish. It might can increase or decrease depending upon. So this is what it's the type of a meristic. Then continuous means it will keep on, means there will be keep on multiple of these things available. It doesn't mean it's keep on increasing, but it's keep on going to the next generation and keep on showing up. For example, substantive, same things, changes shape, size, color of the organism. For example, syndactyly. Syn means together, dactyly means digits. Certain fused digits are present. Okay, then brachydactyly. Brachydactyly is what it is the small fingers present. So sometimes this happens, the small fingers are there, but it might not be present in the next generation or maybe present in the next generation, keep on. Okay, so this is being continuous. Then meristem means increase in the number of organs, means polydactyl means multiple fingers. So maybe if it is a continuous and multiple fingers are there, maybe my kid will also have a similar one extra finger present. Okay, this is about the variations. Next thing in our topic is mutations, which is very important for you to understand. So in mutations are what the sudden abrupt changes that occurs, means we are not expecting, like variations we can expect, but these things are not being expected, and this can occur because of the n number of reasons, and the ones which we can have it called as mutagens. The mutagens could be physical or chemicals, okay, radiations or anything. Okay, heat could cause it, anything can cause it. We don't know who will cause it, but it will happen. It can happen to any part, any time, any direction, or in any individuals. We can't say this. We can't say that I never drink, why I'm suffering from cancer, because it can happen. Okay, it's nothing to lead with, but wait, I am not telling you to go and drink. Okay, don't go and drink and something like that. It just means that of course you will have it, because cancer is because our diet is not that good. Then why we are taking more cancerous product and making us prone towards this thing? Okay, so this is the major point which I want to highlight. Don't take it in a wrong way and then you put the cut clips of these things and put it like this teacher. Okay, Varun, so-called biology teacher and a doctor and not a doctor but a dentist, okay, is telling us to have alcohol and all those things. No, I'm not promoting it at all. Okay, the next thing is involved in the change. Basically what happened, the genes and chromosomes are changed. I told you what is genes, what is chromosomes, everything. So I hope that these things I don't have to tell you again and again. If you don't know, go back, listen to the chromosomal theory, you will understand much better. Okay, so then there are three types of them: gene mutation, chromosome mutation, and genomic mutations. And this becomes smaller to the higher level. Gene is what present on the chromosomes, means one gene is changed. Chromosome means whole of the chromosome can be changed. And genome means whole of the content, whole of your DNA is changed. So this was smaller to the bigger level. So that's why gene mutation is also called as point mutation, because this is the smallest part change that is coming up. Okay, so here what is the change in the nitrogenous base? If you know that genes are made up of what? Nitrogenous waste. If you have done the biomolecules, you know that how the DNA is being made. If not, then don't worry, in the next chapter you will understand, because we're going to be recap it about it. So don't worry about it. So for now, remember that genes are made up of what? Nitrogenous bases. Okay, so there will be change in the nitrogenous bases. Nitrogenous bases are two types, purine and pyrimidines, you know that. Now they are of two types: frame shift mutation or substitution mutation. So frame shift, you have to remember it, because this will come in your syllabus again. In frame shift mutation, what happened? Sequence of nitrogenous base get changed. Now it could be deleted or it could be inserted a new nitrogenous base. So if it is removal of one, for example, if you see here that Ram has cap. So this is, each is representing what? Nucleotide base. Okay. And over here what happened? I delete this. This is deleted. Class S. So what happened? This C will go to the next one, of course, because C will be the continuation of it. So Ram hack app. So means what happened? From here, the sequence changed. And as the sequence changed over here, what happened? There will be what? No proper gene formation. Means till here the gene is okay working. After that it won't be able to work. Understood? Then comes this. Second way is that insertion. You have inserted one or more nitrogenous base. For example, if you have Ram has cap, from there only we starting. First I added what? R. So R is there. Then I added what? E also there. Then I add one more, like for example, Ram has RED. Now the D I have added. Here R added, E added, D added. Okay, and cap. So what happened here? One extra sequence came. This one extra sequence came. This is what? Insertion. So it could be single nucleotide. It is, each alphabet is representing one nucleotide. Okay. It can be more nucleotide or less nucleotide that is added. This is a very important question which later on when we will be learning about the genetic coding, then you will understand. Here you are studying the basic of it. Then comes the substitution. Substitution, what happened? Purine is substituted with pyrimidine. Pyrimidine is with a purine, or anything can happen. Okay. So here what happened? Transition and transversion. Now that transition says that purine is replaced by another purine only. So we have what? Adenine, guanine, cytosine, thymine, uracils. So adenine and guanine is what? They are purine. So instead of adenine, we put guanine over there. So this is what? Purine. Pyrimidine is what? Cytosine, and thymine we have changed it. Instead of cytosine, we put thymine. So these are what? Within the same kind of nitrogenous bases are there. Okay. But transversion is what? Purine is changed with the pyrimidine. Pyrimidine is with the purine. So means adenine is changed with maybe thymine or cytosine, anything. Okay, so this thing you would have understood. This is the easiest one of all. Next come the chromosomal mutations, second one. So chromosome is a big thing. More mutations would be there, more different problems would be there. Now there could be 4 types: deletion, duplication, inversion, and translocation. Now let's just start with the deletion. So in the deletion, it happens what? That there is a loss of a small segment from 1 or 2 chromosomes, and they are homologous chromosomes. Okay, means homologous is same. So terminal or intercalary are the 2 types. Terminal means at the site, intercalary in the mid. If you remember the morphology of flowering plants, we're gonna do this chapter in this year, like this month only, so you would be able to understand it much better. So we have what? Intercalary and terminal. Terminal means at the end. So at the end of the chromosomes, I take this part away. So this part is away. Now the new chromosome is like this. So this is one problem, that we have cut the chromosomes from the end. So this is terminal. Intercalary, if we made 2 different cuts in the center. So intercalary is what? I made this cut. So these are what homologous chromosome, pair of chromosome. Okay, when I'm saying homologous chromosome, a pair of chromosome. Okay, so this is a pair of chromosome. From the mid, I cut this, I take it out and join this. So what happens? CDG. This part is only taken away. The rest is joined back. If you see the second one option. So here what? This. Okay, so this is what intercalary, name itself saying that from where we will cut. Okay, so this is a deletion type of it. Then comes the duplication. Duplication means multiple genes has been represented. So there are three types of tandem, tandem repeats, and displace. So first is tandem. Tandem means, for example, this is a gene sequence A, B, C, D, E, F, G. Now what happens? CD I am repeating. So A, B, C, D, C, D again, E, F, G, H. This is tandem. Tandem means repeats. Now reverse tandem. Reverse means you just reverse the thing. So means A, B, C, D, D, C. So I am reverse, like again random means tandem means repeating the things but in a backward way, in the reverse way. Next is the displaced. In displaced what happened? From a non-homologous somewhere else something came and joined over here. Like A, B, C, P, Q. From where you came but just came. Okay, so these are reverse tandem. Then comes the inversion. Inversion means something which has been flipped. So here what happened? This is your chromosome. So sometimes what happened? We just flipped up like this. Okay, so it goes like this, flipped up like this, and then it make a cut. So this thing joined with this area, this thing joined with this area, and a new sequence has been formed up. So this is what? This is the inversions. Let's just see that it is two types: paracentric, pericentric. So in the paracentric, so what happened? It involves the break in one side of the centromere. Means this is what centromere, either this side or this side. So what happened over here? After centromere, there is a 180-degree rollover. And from there, this cut over here, this area. So if you see that if it is cut like this and this part is joined from here. See, okay, let's just make it. This is cutting from here after joining. And this thing is in the continuation now. So I'm going to be making with a dotted line or maybe with this. Okay. So what happened? This thing joined like this now. So if something is joined like this, so the sequence would be forming what? The sequence would be A, B, C, G, F, E, D, H. So like see that A, B, C, G, F, E, D, H. Okay. This thing you have to understand and practice at yourself, because this thing has been the question and can be the chances of the questions, that when the paracentric thing is happening up, loop is taking place, what will form? Okay, after the cut. Same is pericentric. In pericentric means the centromere, which is the center part, center primary constriction of the chromosome, it is also involved. So can you see that over here? What happened? This thing is involved like this. So what happened? This centromere is also in the part of the inversion. So this happened and then the cut is here. So if the cut will occur here, so means sequence will be what? A, B, D, C, E, F. Okay, so A, B, D, C, centromere, E, F. Correct. So this thing you have to remember, they can ask you. Okay. Then comes the next one, that is translocation. Translocation says what? From one side it goes to the another side. Now what happened? It could be simple or reciprocal. Simple means from one non-homologous chromosome, fragment detach and goes and join with anything else. So this is what? One chromosome. This detaches this part and it goes and join with this area. Simple. Reciprocal means what? This detach from this area, this detach from this area, this goes here, this goes here. So like this. Okay, so reciprocal, opposite takes place. So these are what? 4 types: deletion, duplication, inversion, and translocation. Further, it has the types. Deletion has 2 types: terminal, intercalary. Duplication has 3 types: tandem, reverse tandem, and the displaced. Inversion has 2 types: pericentric, paracentric. Translocation has 2 types: simple and the reciprocal. I hope this thing is understood. Very important for you. Very. Okay. Next come the genomic mutation. Genomic means whole of the DNA, okay, it has been changed. So means here something happens with respect to the ploidy levels. Okay, so ploidy levels, big level things are being taken up. So there are 2 types: euploidy and aneuploidy. This is also very important. Euploidy, what happened? Addition or deletion of one set of the chromosomes to the original. So means 6n is there, we have added one extra set, that is 7n. 6n is there, we have reduced it to the half, 3n, that is haploid. Aneuploidy means one or more chromosome is added. So difference is very narrow over here, that here the set, here the individual. Okay, so here what happened, if 2n is what, if the 2n condition is 46 plus 1 is added, extra one chromosome came up, or it is what, 1 become reduced. Okay, so next this is the types of it. So for the euploidy we have what, 3 types, that is what, monoploidy, haploidy and polyploidy. Monoploidy means single chromosomes only present. Okay, so means if 6n is there, only the n version. Okay, so I'm giving you an example. What happened if there is, if original is 6n, then monoploidy means monoploidy will be only n. Haploid means half of it. Now half of, if the original is 6n, if original is 6n, then haploid would be what, 3n. Now the question is with respect to diploid. In case of diploid organisms, now very funny thing happens. What is the funny thing? Since 2n is the diploid, their monoploidy would be what, n. Mono means single n. Haploidy would also be what, n. So in their case, what happened beta? It requires, it becomes same thing only, monoploidy or haploidy. Okay, so this thing you have to remember. Now the next thing that we have to remember is polyploidy. Poly means more than 2n thing is present. So this is what, multiple copy. It could be autoploidy or alloploidy. Autoploidy means repetition of same genome again. Means this is one genome, same thing, same thing, same thing. No difference is there. So if 2 times is diploid, if you remember that, we used to call AA is what, diploid. If one more time this is coming, we call it what? We call it triploid. If one more after triploid comes, means tetraploid. If 5 times coming, we call it pentaploid, hexaploid, like this, multiple times. And there is polyploidy. Allo means something different is coming up. Allo means allosomes, different zones are coming up over here. So more than one genome is present, like AA was there present in their diploid condition. They take BB also, they take TD also. And this happens in our wheat, trichum, stevia. So the wheat which we are eating is, it has what? It has a different genome present of a different plant, because the wheat which we are eating, it is made up of what? Rye and the wild variety of the wheat. Rye is different plant. This is different plant, but we made this wheat by making first this and then we added one more plant gene also onto it, genome into it. So it is what? Tetraploid. Sorry, it is what? Allohexaploid. Okay, hexa means 6 different genomic chromosomes number were there. Then comes this aneuploidy. Here hypoploidy or hyperploidy. Hypo means less. So means chromosome becomes less. Okay, hypo means what? Always remember beta, hypo word itself means that less. So here what happened? There will be decrement in the number of chromosome. Hyper means increase, addition of the chromosome would be there. Now imagine that original, there is an organism which has what? 4 pairs of chromosomes. If I remove one chromosome, this condition is called as what? Monosomy. Okay, so what it is called as? Monosomy. I reduced it. If I reduce both of them, then it is called as nullisomy. So what it is called as? Nullisomy. And when I remove one from here and one from the other pair, non-homologous chromosome, it is called as double monosomy. Very important for you. Understood? When comes the hyperploidy, in hyperploidy beta, what happened? Addition. So if I add one extra chromosome in this, it is what? Trisomy. Whichever the chromosome I am adding, it had 3 parts of it. If I added 2 into it, we call it tetrasomy. If I added one here and one here, okay, it is called as double trisomy. Tri means 3 chromosomes pair came instead of 2. This is all about what? Your genomic mutations. With this, we have completed today's 3 topics. What were the topics? We completed sex determination, we completed variations, and we completed mutations. Although there seems to be very basic terms, all cramming term, try to read it as much as possible. Of course, this is a cramming topic, but understanding is very important to give some statement based questions. Read it again and again and let me know in the comment section whether you understand it or not. If you like this video, give me a like, it motivates me a lot. Share it with your classmates, with your old 10th classmates also who are struggling to understand genetics. Let's just make it easier for them. Regularly visits for the shorts, which I'm soon gonna be uploading, so that you will always have an on tip certain pointers and you can even excel in your examination. Well, that's all for this video. The notes would be available on my WhatsApp channel link, that is from the Chlorophyll Classes, and even on the Telegram. The description has the link for both of them. That's all. Thank you so much.